Hello guys, this is new. Since we could not meet for class on Friday, instead of skipping our time together and being one class behind, I'm going to instead ask you to participate in our class virtually or online. What that means is that you're going to be listening to my lecture, the lecture you're listening to now. You'll also be taking notes on the notes page that I've provided you. Following our notes in our lecture, I'm going to also ask that you participate and that you take our quiz. We have a quiz. We were supposed to take a quiz. Instead of completing it and turning it into me in terms of paper, you will be submitting a Google form. The Google form will be due before you walk into class on Monday. All right, so let's begin. The lecture today is all about what we're going to say, the cell membrane. And you might ask yourself, well, haven't we been learning about the cell membrane already? And the truth is we have. We have learned that the cell membrane is like a screen or a filter. It's like that screen or that filter we saw using the sand and the gravel and the macaroni, where certain things could pass through because they were small enough to fit through the holes, and other things were not small enough to pass through, and they could not pass through the membrane. The membrane, we said, was semi-permeable. Let's write it down. If I can spell it, that'd be great. All right, that membrane was semi-permeable. However, the cell membranes, actual cell membranes, are much more complex than just a screen. The goal today will be for you to describe the different parts of the cell membrane, as well as to understand a couple new forms of membrane transport or methods by which molecules pass through membranes. So let's take a look. As you can see in this picture, and as we talked about in our bell work earlier in class, is that the cell membrane is made up of more than just one part. In fact, it's made up of many parts. This picture looks quite complicated. However, I want to focus on a few of the more important parts. So first of all, let's take a look at the phospholipid bilayer. The phospholipid bilayer is this bilayer, or layer of two layers of phospholipids. We see one layer moving around this way, and we see another layer coming this way. Right? Remember, and we're going to talk a little bit more about this phospholipid bilayer, but the phospholipid bilayer, again, has two layers. It almost looks like two rows of teeth. In the phospholipid bilayer, we have lots of things sticking in and through and attached onto. All right, some of those things that are sticking through or all the way through the phospholipid bilayer are proteins. As you can see, these proteins come in lots of different shapes and sizes, and they are associated in different ways with the phospholipid bilayer. Some proteins go all the way through the phospholipid bilayer. All right? We're going to call those proteins integral proteins. All right? Those proteins, again, go all the way through the phospholipid bilayer. Some proteins, however, only stay on one side of the phospholipid bilayer. We see different kinds of those proteins, and we're going to call those, instead of calling them integral proteins, we'll call them peripheral proteins. We're going to talk more about those proteins later. You can also take a look from our cell projects. You might recognize the cytoskeleton that are acting as support beams holding up the cell. You also see other kinds of skeleton kind of molecules on the outside. Those are called the fibers of the extracellular matrix. We're not going to talk a lot about those. Again, we're looking at some of the more important molecules in the cell membrane. All right, your job now is to take a look at your notes and answer question 1, or the space in which it says question 1. In question 1, I'd like you to answer the question, what are the two main parts to the cell membrane? All right, why don't you pause? All right, hopefully you've paused and have answered, and hopefully that answer has come to the two parts being, number 1, the phospholipid lipid bilayer, and 2 proteins. Now we've talked about 2 different kinds of proteins in the cell membrane, but generally we've got phospholipids and we've got proteins. Those are the 2 main parts of the cell membrane. Now let's focus a little bit more on the phospholipid bilayer. Again, each of these molecules here is a phospholipid, and so we have lots of phospholipids, and in fact we have 2 layers of them, which is why we call it a bilayer, bi meaning 2. Alright, in this phospholipid bilayer, each phospholipid has, we're going to call it a head, and 2 tails. Just like any lipid, the tails that we're talking about are fatty acids. Okay, and like any fat or oil, fatty acids do not like water. They are afraid of water, and as a result, they will be called hydrophobic. So this area over here in which we see all of the fatty acid tails are going to be recognized as hydrophobic tails. Right? They do not like water. And they end up pointing to each other, and inside the cell membrane is because the outside of the cell membrane, the extracellular part, and the inside of the cell itself, the intracellular part, or the cytoplasm, both of those are watery environments. Remember our potato experiment. The solution that we were working with was a water-based solution with some sugar, and the potato we know had water in it as well, right, inside the potato. And so both inside and outside of the cell membrane, we've got watery solutions. Fatty acids do not want to be around watery solutions, and so they will try to hide from them. So that's why we see the fatty acid tails pointing inwards from the cell membrane. The heads of the cell membrane, though, the heads of the phospholipids, are made up of phosphate, and phosphate is an ion, and ions are hydrophilic. Right? So we've got, on our phospholipid bilayer, we have hydrophilic heads, and we have 2 kinds of hydrophilic heads, one on each layer, and then we have hydrophobic tails. So, in the space that says drawing 1, I'd like you to draw a phospholipid bilayer. Do your best to draw the phospholipid bilayer without actually looking at the picture. Try to do it from memory. I also want you to label the hydrophilic area on the bilayer and the hydrophobic area. Please pause and start drawing. All right, hopefully you've paused and have drawn your phospholipid bilayer. I'm going to draw my representation of a phospholipid bilayer just so that we are clear on what we should be drawing. A phospholipid bilayer can be so simple as just drawing some circles, drawing tails, drawing more tails. Remember, we've got our hydrophobic region, and drawing some more circles as the hydrophilic heads. Again, the heads will be hydrophilic, and the tails will be hydrophobic. And again, like we said earlier, we have different kinds of proteins associated with the membrane. Those membrane proteins do lots of different jobs, but in many ways, a lot of times those membrane proteins simply act as doorways. In general, we can classify the membrane proteins into 2 kinds of proteins. Again, like we said earlier, we're going to talk about integral proteins, proteins that pass all the way through the cell membrane, and peripheral proteins, proteins that stay on only one side of the cell membrane. So quickly, you can answer question number 2. What is the difference between integral proteins and peripheral proteins? Why don't you pause and answer this question? All right, hopefully you've answered that question. And again, to emphasize the point, peripheral proteins, let's just call peripheral proteins, only on one side of the membrane, whereas integral proteins go all the way through the membrane. Certainly, you might have also said that the peripheral proteins were a little bit smaller, the integral proteins a little bit bigger. Maybe you said that the integral proteins were more vertically aligned, whereas the peripheral proteins were more horizontally aligned. Those are all very good answers. The important points, however, are of whether or not it goes through or it stays on only one side of the phospholipid bilayer. All right, now we want to talk a little bit more about membrane transport. And we want to talk about methods of transport that are different than diffusion and osmosis. Now, we've studied diffusion and osmosis quite a bit with our lab and our activities. So let's review what diffusion is at least. Let's remember that diffusion is the movement of molecules from an area of high concentration to an area of low concentration. What we can do is we can imagine that when molecules are diffusing, they're moving in some ways analogous to the motion on a hill. Let's imagine that we have a boulder, and that boulder is at the top of the hill or near the top of the hill. It is at a high concentration. If that boulder is released, we don't have to do anything to that boulder because that boulder is going to fall. It's going to move without much of our help. Right? We don't have to put any energy into it. It's going to move towards low concentration. So all molecules want to move from high concentration to low concentration. You don't have to do any work to make them move. Just like you didn't have to do any work to make the iodine move across the bag. You didn't have to do any work for the water to move out or into the potato. It happened on its own without any energy. And this is what diffusion is. However, remember that the phospholipid bilayer can only allow certain things to pass through. Other things that might be too big won't be able to pass through. Molecules like this molecule, those yellow molecules that you're looking at on your screen. Those yellow molecules are way too big to pass through our phospholipid bilayer. But they can still diffuse into cells. They do so using what we call a channel protein, or any kind of integral protein. Remember that integral protein is on both sides of our membrane. And this channel protein simply acts like a doorway, an open doorway, and anything that is small enough can fit through, and especially things that are too big to pass through the phospholipid bilayer. And notice that these molecules are at a high concentration outside the cell and at a low concentration inside the cell in the cytoplasm. And so the movement of molecules is moving from high concentration to low concentration, just like any other kind of diffusion. However, the diffusion is facilitated, or it is helped by this integral protein. That's why we call it facilitated diffusion. And just like any diffusion, it does not require any energy out of us in order to accomplish this. Here's another example of facilitated diffusion. In this example, we see glucose, a really important molecule if our cells are going to generate energy. All right, but glucose is, again, too big to pass through the cell membrane, to pass through the phospholipid bilayer. It needs an integral protein. However, this integral protein, instead of acting like an open doorway, sort of operates like certain locks operate. All right? And the glucose molecule operates like a key. And so once the key fits into the lock then changes shape. And once that lock changes shape, it then releases glucose into the cell. Once glucose has been released into the cell, it can then change shape again, or recover, and then repeat the process over and over again. The key can fit into the lock will change shape, will release the key, and the lock will change shape again. Again, this does not take any energy. It is just a protein facilitating or helping the diffusion of a molecule like glucose. Here's the definition of facilitated diffusion. It is the diffusion through integral proteins of large molecules that cannot fit through phospholipid bilayers. Please write this down in your notes. And now what I'd like you to do is to draw an example of facilitated diffusion. You may draw this example as a channel protein, like an open doorway, or you may draw an example like the lock and key model of glucose transport that we talked about. This you might have a drawing that looks something like this. We can talk about this is our channel protein that acts as a doorway, an open doorway, and we have our phospholipids. Oops, I'm sort of being lazy here. Don't mind me. Okay, phospholipids, phospholipids. Here's our bilayer. All right, sort of a sloppy bilayer. I'm just going quickly. All right, I'm going to try to use a different color here. Let's see here. Let's use green. Okay, and so we've got molecules on the outside. Those are lazy molecules too. All right, molecules on the outside at a high concentration. We've got a couple molecules moving through and a couple molecules at low concentration. The movement of molecules will go from high concentration to low concentration. That's the facilitated diffusion. All right, so let's revisit our definition of diffusion. Remember, diffusion is moving from high concentration to low concentration. And if that boulder might be falling down, we don't have to do anything to help it. However, what if we had that boulder at the bottom of the hill at low concentration and we wanted to move it to high concentration? We'd have to push it up the hill. That would take a lot of energy. You might feel really tired after doing that. Right? So any kind of movement where we want to move molecules from a low concentration to high concentration, it's going to require some energy. And we might want to do something like that if the molecule, for example, is something that we don't want in the cell anymore. We want to pump it out, and we don't want it there, any of it at all. For example, there are points in the cell's processes that wants to take hydrogen ions and pump them out of the cell. In order to do that, we again use a integral protein. All right, and that protein acts as a pump where it takes molecules from low concentration and pushes them out and towards high concentration. We're literally trying to pump all of those molecules out, but in order to do that, it requires energy in the form of a new molecule known as ATP. We're going to talk a lot about ATP in the coming days. And so a definition for what we're going to call active transport is the movement of molecules from low concentration to high concentration. Right? Again, from low to high against the gradient, or we can say uphill. All right? And any kind of active transport uses ATP, or uses energy. And usually we're using energy to push or pump molecules through integral proteins. Our final drawing asks you to draw an example of active transport. Again, we should have an example of a membrane with a protein, an integral protein inserted in, pumping molecules from a low concentration to a high concentration. And that concludes our lecture. Now is your time to start working on your quiz. You're going to submit the quiz again through the Google form, and that quiz will be submitted before you walk into class on Monday. All right, have a great weekend, guys, and I'll see you on Monday.